Alright guys, welcome to another video for Falcon Biology. In this video, I'm going to be looking at the different slides that you're going to be seeing in class under the microscope. So basically what I'm saying here, I'm going to be saying or explaining in class as well. So this is just to help you in case you missed something in the lecture or maybe you just need something explained over again. So we're taking a look at the different types of bacterial cells that you're looking at under the microscope, and they basically are differentiated by their structure on the outside layer of that bacterial cell. Keep in mind that every cell does have a cytoplasm or a phospholipid bilayer, just like the cells in the human organism. Now, where the bacterial cells differentiate is that, as you can see, there are extra layers on the outside, kind of like a cell wall in a plant. So here I have an example of a bacterial cell on the right, and on the left a different bacterial cell. So you'll notice on the left that there's a very thick wall called a peptidoglycan wall. This is amino acids with glucose molecules. This particular peptidoglycan layer aids in giving the bacterial cell extra protection. It also gives it extra strength, structural strength, helping it to withstand the external environments. Now, if we compare it to the bacterial cell on the right, you'll notice that the peptidoglycan layer is much thinner. It's about 10% of the overall layer, whereas on the left it's about 40 to 60%. So a much thinner layer, but it also does have that extra outer membrane layer, okay, for extra protection as well. And also the outside, this particular layer does have other antigens on the outside that are toxins to humans. So you can get nauseous or you can get vomitus if you do have an infection with this particular type of bacteria. So we essentially have two classes of bacteria. Now, why is this important? If you go in with strep throat, one of the first things they're going to do is they're going to want to swab, get a sample of the bacteria that's in the back of your throat, and they're going to want to stain it. Okay, so this is a staining process that's going to help differentiate or help the doctor figure out what type of bacteria you have, because based off of that, they're going to then be able to determine what kind of medication to give you. So the first stain you're going to get is a purple-violet stain. And if, in fact, the bacteria you have has that thick layer of peptidoglycan, okay, those crystals, those molecules are going to get absorbed into that very thick layer. And as that absorbs that purple, it will stain the bacteria purple. So when you look at the microscope, you see these. You see the different bacilli and you see the cocci that are stained purple. Taking a closer look at these, you'll notice also that they're clumped up and they're organized in a specific way. On the left, you have streptobacillus. Okay, strepto meaning they are aligned in a chain-like fashion. So anytime that you see bacteria lined up in a chain and a line, then that's strepto. Now, whenever you see them lined up in clumps like grapes, that's staphylo. Okay, it's like a staph infection or streptococcus or strep throat. Okay, now let's say that you stain it purple and it did not hold. So it didn't, when you washed off the stain, you had nothing there. So then there's a counter stain that is basically one that will contrast the stain that you just did. And this one is absorbs pink. And that's because of the outer membrane. So this one will absorb pink, and you end up with bacteria that are, again, pink. And this is called a gram-negative bacteria. And again, you still have the same basic types of structure. You still have bacillus, you still have cocci, you still have staph, and you still have strepto. But the difference, again, in these two different classes of bacteria are in the outer layers. And depending on what type of outer layer it has, it will either be a gram-positive, which means it absorbs the initial stain, the purple, or if it doesn't have the thick peptidoglycan, then it will not absorb the purple, but it will absorb the pink, being gram-negative. Okay, so here again are the visuals of what you're going to see in the microscope. So understanding what you're looking at, I think, will go a long way in helping you be more efficient and effective when you're in the microscope. So at this point, what you really need to do then is practice practice manipulating the course objectives on the microscope, and the fine tuning of the focusing on it, so you can actually get these images on the microscope, because those can be pretty challenging, but it's just a matter of practice. Okay, so I think that'll do it for this particular topic, and we will